Hello everyone, my name is Vinayak Deshpande. I shall be presenting my project on image segmentation using k-means clustering algorithm. This is as a part of my EEE6512 course called Image Processing and Computer Vision, which is under Dr. Dapeng Oliver Wu. Let's go through the agenda. We shall start with what exactly is image segmentation. Then we'll go through some of its application, and we'll see some of its existing image segmentation methods that we have, and we will go through the proposed k-means clustering algorithm, and then we'll see how the block diagram shows the entire process of this process of implementation of this algorithm, and we'll go through some of the results, and we will have a small demo, and we'll conclude this project report. So what is image segmentation? So this is basically partitioning of image into multiple meaningful segments or partitions. So when I say meaningful, it means that it should be easier to examine. When we segment images into different segments, we want to study each of those individual objects and maybe use them for post-processing. So each partition here could be a set of pixels which share the common property. For example, pixel intensity. So a group of, or a set of pixels that have the common properties such as pixel intensity can be grouped together or segmented together to form a partition. And what exactly is the application of this? So we can use this segmented or partition objects in object recognition, or we can use it even for image compression, or we can use it for image editing or database lookup. For example, if you have an object, you can easily look up into a huge database and recognize it. So one of the major applications is in medical field. So we can use it in brain MR imaging. For example, we can see here the brain MR imaging where there is a tumor which can be easily detected using this image segmentation. We can also use it for blood cell image segmentation. For example, in patients with leukemia, the blood cells not only contain the red blood cells but also contain infected blood cells. So we can easily detect such blood cells using this image segmentation. So what are existing methods we have for image segmentation? There are actually a lot of image segmentation methods that have been in place for a decade or so. But I have listed some of the important image segmentation methods that have proven to give most efficient results and most accurate results. The first one is called the graph-based segmentation. This algorithm assumes each pixel in the image to be a node in a graph, and lines connecting those pixels to be the edges in a graph. So basically, in this graph-based image segmentation, we see the image as a graph that contains nodes and edges. The second one is the watershed transformation. This method takes into account the gradient of the desired image to be segmented, and this works independent of the shape or size of the object. But the major disadvantage of this algorithm is that sometimes this produces over-segmented output images, and this could be because of the noise present in the image, or could be due to other irregularities in the image. Let's see the third one, that is region growing. Region growing is in this we begin with one pixel of a potential area of interest. So we decide one particular region in an image, and we start with one pixel, and then we try to grow this by means of adding up adjoining pixels until we find the similarity between the pixels being compared. The fourth one is binary segmentation, or also called as the histogram based segmentation. in this, the idea is just this algorithm works most efficiently for very simple binary images. So in this particular object consists of pixels of same grayscale values. So we take this advantage of binary images that a particular object will have almost the same pixel intensity values. So based on that, we actually segment the images. And the final one is K-means algorithm, which is actually the proposed algorithm for this project. So I'll be explaining it in detail in the next slide. Okay, so what K-means clustering algorithm is? For example, if we have a set of observation points given by x1, x2, x3 and so on till xn, we want to have k sets of clusters which are denoted by s1, s2, s3 and so on till sk. So all we want to do is take each of these points and put them in each of these clusters, and each of these clusters will have a predefined centroid which is given by this equation. So the idea is we want to minimize the within cluster sum of squares, also called as the WCSS, till the point where it converges. So for example, consider the leftmost image. We have 3 points ABC here and BDE here, and we initially defined 2 centroids, and in the upcoming steps we start assigning each of these points ABC and DE to the respective centroids. So we in every step we calculate a new mean value, new centroid value until we reach a convergence point. So in the block diagram for the algorithm, we initially have an input image, and then we define initial centroids for this image, and then we assign data points to each of the clusters. For example, if we have k clusters, we move the data points to each of the clusters based on how far they are from the centroid, and then we see if the desired convergence is reached. If no, we calculate the new centroid and assign the data points to the cluster. If yes, that's the completion of clustering, and we finally have a segmented image as shown here. Okay, so let's evaluate some of the results we have got from this project. So this is my original image, and this is my segmented image. So I have set the number of partitions to be 4, which means that all the colors we see on the right hand side, in the right image, we have 4 different colors that are actually the mean of all the colors present in the original image. So if we had set the number of partitions to be 5, we would have had 5 different colors defined in this image. So in this, the first image is the same as the previous one, that is segmented image, and on the right hand side we can see 2 more images that are actually the extracted images of the original image. These images are very significant in that we can see that if the label is 1, it is only showing the pixels that are related to this particular color. So depending on the number of partitions we assign, there will be those many number of labels. So we can individually print those labels, which means we can individually have those objects shown on the picture. So if I set the label to be 3, my the algorithm will print only this image which has all the green colors in it. So this algorithm or this function that is used for extraction uses 3 main parameters, that is the original image that is used for segmentation, and the image that we have labeled. So we when we get segmented image, that will be labeled image, and then the expected label value, which is 1, 2 or 3. So in case of 4 segments, we will have 4 different labels. Similarly, we can do it for grayscale images. This is my original image, and this I have partitioned into 5 different segments, and this is the extracted image. So we can see that when I set the label equal to 2, I can clearly see the tumor here. So it the number of partitions, it really depends on the kind of application that we have and the kind of image that we have. So we can set this number to any value and see different results. So before I go to conclusions, I want to show you a small demo. Okay, so this is my source code. So I have set the initial number of partitions to be 4, and this is where I'm actually inputting my input image. So if I run this, it shouldn't take much time actually. Okay, there seems to be some small problem. Okay, so we have the original image here, the segmented image, and the extracted image. So we can see that the label being set here is 3. If I change the label to 1 and rerun it, okay, so that was my previous extracted image, and this is my new extracted image. So we can see that based on the label number we can get a different extracted image. This is very important because if you want to track a particular object, and if you want to see how it's moving or how it's behaving, then it's really important if we have something like an extract function that only returns you the extracted image. So coming to the presentation, so in general the conclusion is image segmentation basically simplifies the image representation, how we represent the image, and this is very important for post-processing, for example object tracking or image editing, etc. Extensively it's used in medical field for cancer treatment and tumor detection. So I have actually shown you one example where the tumor was detected. And the best thing about K-means algorithm is that it runs in linear time due to better heuristics. Usually if you increase the number of segments or partitions, the complexity of this algorithm will be very high if you set the number of segments to be a large value, but better heuristics make it to run in a linear time, and its performance, as I said, depends on the number of segments or partitions. Okay, so here are the references, and I would like to thank Dr. Dapeng Oliver Wu for providing me with this opportunity to work on this interesting project. So he has been really helpful. Thank you. If you have any comments or questions, please please put them in the comment section. Thank you.